So welcome everyone to our last panel of the day for this track. Please, if you haven't yet done so, change your name and your preferred pronouns on our naming convention. Let me just present here. So this information to keep the conversation going, the Slack channel has been going really, really strong on the art and design track. So let's keep it there and let's have the links and everything that you want to share on there. And thanks to our sponsors. And we're going to start working with talking about this prototyping the future of biomaterials. And I'm really interested in this. I had the chance to talk to all of the panelists beforehand. So I got a sense of what we were going to develop here. And what I found really interesting is like, this is gonna sound weird, but it's not like a traditional way of approaching biomaterials. And I think this is something that is going to open up the conversation. And most of the panelists are gonna be talking from a point of view of a process and in different spectrums of biomaterials, not just the material itself, but just using the materials as an excuse to talk about things that are very tangential and about our behaviors, culture, society, and how we put these materials into work. So it's more like a reflection panel. And we have, I don't know if who you're seeing right now. We're gonna have these amazing speakers. So Kai, she's from the Biomimicry Program Specialist in the Living Lab Montreal in Canada. I'm gonna let you guys present yourselves in more depth in each one of your talks. I'm just gonna give your names and affiliation. Orkan, Telhan, we had an amazing talk with him about biomaterial soil as a biomaterial, but in the other side of the spectrum, like in the degrading part of it, which is really interesting and thought-provoking. Laura, she's from Mexico and she's living in Russia right now. And we talked about also about the differences and of resources and how she's being a designer, being the artist within the lab where she works at there. And Dilan, she's working on her PhD, working with biofabrication from mycelium. And actually she went into fungi and just using the environmental stimuli to kind of like a free way of growing fungi instead of being more like a gut approach where I give you the shape. Is more collaborating into a gardener type of thing. And I'm gonna be moderating. I direct our research group here in Buenos Aires that we're working on biofabrication with mycelium as well. And we're most interested mainly on the process. So I think this is a panel for a process. Please keep your questions going on the chat and we can ask the questions to our speakers as we go along. So let's start with Kai. I'm gonna stop sharing my screen, Kai, and you can share yours. Let me just find this. Hold on. Can you just share your screen? It would take mine out because I can't find the panel. Oh, here it is. Great. And
sorry. Yeah, I'm trying. Sorry, I'm getting there. Okay, and I'll play. So hello, everyone. Thank you very much for giving me this opportunity to present something very fermenting, but prototyped. I also have jumped into the cold water with this. I've taken the first time ever to formulate this for the Bio Summit, and it was my means of application to get here. Sorry. So I'm going to go one step backwards here. Just start again. So my question is, do we need new shape imaginaries? This is for me an example that's done by Jenny Sabin, which illustrates a little bit what I'm trying to get up with the project I will present, where I'm asking the question of social acceptance of biomaterials, and I'll situate a little bit how I got to this stage in my experiences. First of all, I used to be a clay artist a long time ago, nearly for my life, 25 years ago. In the top image here, you can see me work in the heat beat and treat process with a kiln behind and obviously a large footprint. And a couple of years since then, I've started or tried to experiment in a very sort of home fabrication way with biomaterials, more also to teach my children in circularity principles, because I'm also the local rep for the Circular Economy Club in Calgary. And what I try to do with those projects is trying to incorporate a little bit of the culture of an individual household, use household wastes, and work my way up to workshops I could do, for instance, in schools or something, where we would respect our culture. In our family, it would be mussels. We have French background in my family, and so we have a lot of mussel waste, and I was looking to try to process this, and that was one of my big failures, where I didn't have access to tools and I couldn't sort of move my way forward to any type of finished product. Second attempt, or another attempt of many attempts, was to do some leather-like material for a school project with my son, and the outcome was something more like plastic, and so I have a long way to go. And those were my individual prototyping experiments, and now I will later on still experiment with others, which I will talk about later. But to situate where I'm coming from, I've witnessed, because I've been a lot involved in the local startup scene, and I've witnessed a lot of other failure, or let's say blown out of proportion expectations. So my critical eye is on design speculation or hype cycles and technology readiness, and especially our cultural readiness for this. So I've been witnessing that with carbon upcycling, who is using carbon sequestration technologies with fly ash, for instance, for concrete, but also for other products. And the hard time and the hard road to commercialization and industrialization has been very much part of my experiences of the last couple of years. So, and also in the biomimicry community. So based on this, I've been feeling this project which grew inside me, where I'm thinking about how do we prepare society for a different kind of aesthetics. Heidi, I took this photo from you, didn't ask permission, so I'm doing it now, just to give a little bit of examples, which most of you know much better than I do, that moving away from what we're used to, the rectilinearity, as Margaret Wertheim from the Institute of Figuring calls it, of engineered products which have a perfectionist line and structure and are solid and based out of a cultural history, out of the Western Platonic sort of tradition of Platonic shapes. I'm trying to find ways to educate towards standardized and non-standardized materials and variability, colors which are less shiny, which are maybe matte and from smooth to solid and sleek, so rugged materials. And I'm wondering what or how we could move forward with something like that. So what I've been fascinated about, and for me a move back to my own cultural roots, is the work of Friedrich Froebel, and a lot of what I'm presenting here today in this slide is actually something I only learned today. So I'm asking the question of, could we find some sort of Froebel gifts which would allow us to, at a very basic or fundamental level, create tools to educate very young children or society for different kind of aesthetics. So why am I saying this? Because Froebel based his pedagogy, and he's the inventor of the kindergarten, for all those who don't know. He based his gifts, which are the playful tools he uses in the late 18th and early 19th century for kindergartens on the molecular seed of animals and plants. So he has a whole pedagogy behind it, and he focused his gifts on three types of forms. So the forms of nature or life and things of the world, forms of beauty, so that's art, and forms of knowledge, science, mathematics, and geometry. So you have a couple of examples to the right. How am I doing for time? Sorry, I didn't launch my timer. Sorry, and I can't move forward with my, okay, so there's another example of this here. And if you would like to read up on this, I would really recommend the book by Norman Brosterman, Inventing Kindergarten, because he traces the effect of this movement, because that was also a movement and a social movement for the kindergarten in the 19th and 20th century. He traces back a lot of the influences in abstract art or geometric abstraction to Froebel's gift. And so I'm asking the question, could we collectively come up with a new kind of Froebel gift, which would allow us to move social acceptance of biomaterials, which differ widely from engineered and industrial materials? Could we find ways to move this forward? And on the educational or conceptual side, I'd like to move or ask questions which go beyond that, because I'm also criticizing, I think, the purely STEM-related models of game and play we have, or scientific STEAM and play, kits. An example, the last one is, for instance, Lux Blocks, which try to imitate nature. And in the end, also comes back to something which is very static and very engineering or mechanistic mindset, or it's mathematical, like Zoom tool. Minitubers is better for sure, but so these are questions I'm asking with respect to that. Now, what I've been trying to do, and I want to show in the last part of my presentation, is how I use, for instance, the inspiration of the structure of bone to get to what I feel is more organic forms, and hopefully some type of gifts which could be used to introduce intrinsic architectures of biomaterials. So bone for me is excellent because it's at the molecular level combines two opposing and contrasting materials, so collagen and hydroapatite particles. So the hyperflexible collagen with a rigid hydroapatite. And for me, this has a symbolic meaning of how you could combine in a special sort of spatial combination or arrangement these elements to work together. So I've got my materials in here, but without an example, it doesn't mean a lot. So I'm just going to move forward. So this is something how it can look like. That's when I use masking tape, because I'm still looking for patterns and forms there. So I'm in prototyping mode. And I use masking tape because it allows me to aggregate forms. It's sticky, I can mold, and it has a it's very democratized. So I'm echoing Manu Prakash from this morning with respect to democratizing access to tools, and which is also why I'm arguing against this type of science kit, and it echoes also Froebel's thinking. So my work, I'm moving forward myself, thanks to having met last week Martin Dade Robertson from the HBBE, and I think Dilan, you know him very well. So I read his book, Living Constructions, and I feel it echoes a lot of my work, and I feel I can move forward with a lot of the thinking I found in his book. So I'm very happy about that, towards what I hope to be cell landscapes or epigenetic landscapes. And this is just the beginning. And I'm looking for anybody who would be happy to collaborate, especially scientists, on this with me. So the point I'm looking to is, at the fundamental level, I would hope that we as a society would embrace something like Froebel gifts to educate hopefully beyond the metaphors we have right now. And I'd like to end with a quote from Tom McKeek, who is the founder of the Zygote Quarterly, which is our biomimicry journal, where I also echo what you just said, Heidi, in the beginning, that we're, I think, modifying the way we do things and the professions we exercise by becoming nurturers and cultivators and gardeners. So thank you very much.
Thank you so much, Kay, for your talk. We had an amazing talk last week, and all these concepts that are surrounding this conversation are great. And this is what I mean, what not so much about the traditional way that we're thinking about biomaterials, but biomaterials as a way to reflect on these topics and maybe just change the material itself in which we're talking about. So now I'm gonna leave the room for Dilan.
So hi, I'm Dilan. I'm a third year PhD student in Newcastle University now, and I'm going to talk about my PhD work. And you know, when I start looking at biomaterial, looking at biodesign projects, I realized that the organisms are fabricated through a supporting medium, such as a mold, a scaffold. And I was always like naively thinking biomaterials have a metabolism, so they can actualize reproduction, growth, assembly, adaptation, or decay on their own in response to the environment. So I was thinking using a traditional molding technique always reduces the degrees of freedom that the living creatures have, because the materials lose their inherent potential when they're cultivated in a predefined form. But after I've started my PhD, I realized that all organisms cannot be formed without a supporting structure. And every biomaterial has like different requirements to grow and form. So some of them cannot be fabricated without a supporting medium at all. But among other biomaterials, fungus can autonomously form 3D structures, and they have high plasticity. So I started to work with fungi, and within my research I aim to go beyond the mycelium brick and use fungi as an active material instead of an obedient one. And in this presentation I will try to demonstrate a material making process through showing my process. So my question is to answer how to guide a living material that has its own tendencies and grow it with minimum intervention. And I will question what's the role of a designer in a system which is not fully under their control. So there's a spectrum in the level of control, and it can be like entirely regulated, or it can be fully liberated. So plants growing freely in the nature is the most extreme side of liberation. Mechanical controls such as 3D printing or CNC milling of materials, they are mostly the extremely controlled version of the production. So in my study, I'm aiming to take fungus and trying to slide it through to the more liberated, to the more controlled side from the liberated one. But if, you know, when you say I'm going to control a material, a living material, but how do I do it? Am I trying to be a god or a gardener? It's going to be my question. And because, you know, God has like complete control over growth, but gardener works with the elements and tries to push the growth to a direction. So I'm aiming to be a gardener in my projects. So I started working with mycelium just to understand how to form fungus. So I did some extremely basic experiments just to get the organism a bit better, because I am an architect and, you know, I have no biology background. So I just try to understand how to inoculate seeds for my future tests. I try to understand if agar amounts makes a change in the growth in short term or in long term, and what's the oyster mushroom's preferred habitat. And at the same time, I try to look how to block growth sharply, because stress sources can be used to carve mycelium biomass at some point, at some certain points. So all of these preliminary experiments helped me to formulate the growth of oyster mushrooms for my next steps. So I tried to merge the information from all the tests and come up with the idea of a waffle structure. So I generated different pots with high to low nutrient level for mycelium to follow and grow. And I was assuming it will grow in its favorite mediums, and it will grow less in its less preferred ones. So my dream was to develop a reproducible system. You can imagine this like you can imagine like the growth will be through a deposition of nutrient, like an inverted 3D printer. And at the beginning, my system worked, but after a while, the growth stopped and my mycelium began to wilt away due to dryness, and because there was a decrease in the nutritional value. So I realized that I need an irrigation system to prevent that decline. So controlling the growth by feeding mycelium from specific points continuously can be used as a method to format, and each cubicle can be imagined as pixels generating the overall geometry. Because more receiving food parts will grow more, so the mycelium will start to rise from the plane. But after a time, the increasing number of cells in favorable nutritious cubicles may reach their point that they cannot show any more growth because there will be a crowding. So there's a need to have a scaffold for the mycelium to cling on. Otherwise, the system will reach a point where there will be a flip due to the overcrowding. And another problem with this system, this mechanism, is that it only feeds mycelium in one direction. So mycelium will be only extruded through a horizontal plane. And so as an architect, it will be difficult to make changes in the section. So it will be limited, and it will be limited in any changes in section plane. So after I saw that all these limitations, I found that growing mycelium, bottom-up approaches, it's difficult, and molding is better for mycelium basically. But a tessellated agar system also can be digitalized with an irrigation system, but the system doesn't require a digital control that much. And mycelium doesn't have that much 3D potentials also. But when I look at the mushrooms, the fruiting bodies, they can give really complex 3D structures, and they have obvious morphological difference. They are more adaptable to a digital system. They exhibit more varied forms. They're more plastic. And that's why I continue to work with mushrooms rather than mycelium. And it's still the same organism, but a different part. And from now on, I try to understand how to form mushrooms. So physical environments such as temperature, humidity, CO2 level, they all have an effect on mushroom morphology, and almost each part of mushrooms can be modified by changing these parameters. So I decided to form the fruiting bodies by controlling the environmental conditions that they live in. So, but you may think why? Because mushrooms are not useful as a building material, and they cannot be an actual building material. But my question is not if it's useful or not. I'm just trying to understand how form comes about and how to influence the morphology. For example, in here, these are same type of mushrooms, except the CO2 levels. Every environmental condition was the same. So as you see, they took really different shapes. The morphology of both stipe and the cap, they changed a lot. So for that purpose, I designed a chamber that establishes the conditions for the growth of mushrooms. So this is a robotic growth chamber. It's composed of Arduino, it has sensors, there's an algorithm, and there are devices like humidifier, heat bulb, light source, and fan. And I'm happy to call it a robot because a robot can sense, think, and act. So my chamber can do all of them. So the designer can control the reactive behavior of the chamber digitally through a code. By the help of the sensors, I can measure the growth of mushrooms, and this provides a feedback for the control. So it helps to create the inhibit or promote growth. So the influence on mushrooms on the growth chamber creates a feedback loop. And the fruiting bodies as an outcome become an input to, to change the system, to manipulate the system. And I built 3 of them because, I wanted to conduct a controlled experiments. And you may think why there's a need to have a digital interaction? What's the benefit of digitalized tools? In my mind, digital making helps to work interactively, and it's a collaborative way of making. And it will be interactive because the designer will influence the environment, and the designer will influence the organism accordingly. So I recently started to use my growth chamber, and now I'm running some experiments by changing the variables one by one. And I'm observing individual mushroom morphologies. And for example, this is what I reached, changing the humidity level, but I'm still working on creating a surveying method for my future tests. And as you see, mushrooms are adaptable to a digital system, and they are reacting to the digital interaction because they are highly responsive to the environment, and they can create complex 3D structures without a mold. And as the next step, I'm aiming to make a catalog of mushroom grammars, and it will be a kind of a manual that helps to build a dialogue with fungi. Because there's a...
Dylan, sorry to interrupt you, but we're running out of time.
Okay. So yeah, this is my future goals. Yeah, that's all.
Thank you so much for your talk. I think it's super interesting about the humility you need to learn from an organism. And you said when we talked, how naive you were at the beginning, just thinking you could control all the variables that our organism needs in order to grow. Yeah. And I think this working towards something more collaborative, it's really interesting. So now I would want to give the room to Laura. And Orkan, I left you for the end. I know you were before that, but I want to close up on this soil and how this decomposing of materials comes about. No Thank problem. You, Laura.
Okay, do you hear me?
Perfect.
Great. And can you see my screen? Okay, so I will be talking about the use of biomaterials for the sake of art, contemporary art. So I have 3 main points. One is how artists are working with biomaterials, how artists and scientists collaborate for science art, and the 2 examples, how to overcome the difficulties to present biomaterials in the galleries. So a little bit about me. I have a background in biotechnology and molecular biology, but after a wonderful crisis of not finding myself in academia, I decided to come to art and science. And then I found this place in ITMO in St. Petersburg that is the first Master of Art and Science inside a technological and scientific institute. So we have kind of advantage to getting inside the labs and collaborating with scientists. But at the same time, we keep up with a strategy of do it yourself. Because when we look for the environment of growing biomaterials and then put it in the gallery, do it yourself is troubleshooting a strategy. So we understand what needs to the materials has if we try to accommodate a classroom and we do a workshop there. So we work with bioplastics from cornstarch, bacterial cellulose, and also, for example, a slime mold that is very popular now in science art, Physarum polycephalum. So for example, in this workshop, we were just in a normal classroom working with Physarum, and actually to make our area a little bit more sterile, we put some candles. And we really were like these 5 women in black with candles. It looked like we were making some witchcraft. But this ambient or this environment inspired one of the artists taking this scientific workshop to create artwork working with Physarum, but we couldn't bring the microorganism into the gallery. So she created an analog Physarum, a digital Physarum, taking into account the movement of the slime mold, and then with a tracking image technique, we could grow it in a scan. For example, you can put your hand in a scanner and then Physarum was growing there. And she decided to make this archived machine as a Physarum fortune teller. So the people visiting the exhibition they were putting their hand, Physarum was growing in the lines of the hand, and they receive ironic and kind of absurd prediction of the future. What they didn't know was that there was a hidden camera taking a picture and connected to a face recognition app that was looking for them in the internet, finding the Instagram or Facebook, and then also giving some data in this nonsense prediction of the future. So the statement of the artwork is about our trust in other microorganisms, the trust in technology, and the awareness of doubt of all the data that we find in internet, the fake news, and all these absurd predictions sometimes. This is a way we could bring something from the lab to the hands of the artist, and they transform it into a digital platform or media to exhibit in the gallery. But there is other approach when the artists come to the laboratory. And in this case, it was a project that I did, although I have the background in biotechnology, my role here was as an artist. And I collaborated with scientists working with synthetic membranes. And I decided to bring something to the lab that is very famous now, also the bacterial cellulose called kombucha. And in Russia, they call it Chinese grip, that if we translate it to English means Chinese mushroom. And people was very surprised to know for the first time that it's actually not a mushroom, but it comes from a community of yeast and bacteria. And this layer is a bacterial cellulose layer. And what I propose to do is to culture it with nanoparticles of titanium dioxide, that they are photocatalytic. And to culture kombucha, we only need the tea, water, the source of carbon, in this case, a lot of sugar. And we put the nanoparticles in the tea. So because the bacterial cellulose grows in layers, the nanoparticles were trapped in the matrix of the bacterial cellulose. And we start to make some experiments. By this time, I already was feeling, and for all the previous projects with kombucha, that this really looked like skin. And I started to think how the light was affecting my own skin. And in our experiment, we put the membrane in a chamber with a solution with ions, and we start to see that it reacted to light. Interestingly enough, the ions migrate against the gradient of concentration. So I was very excited talking with the chemists in the lab. They were saying that it could work as a capacitor. And then I was thinking, okay, we can connect some millimotors and make it vibrate, and we can make a skin suit. That's why the project was called Skin Futurism. And then pandemia came and isolation started, and my project was not considered as a primary work in the lab. So I was sent home with all my kombuchas. I put it in my kitchen and I started to grow them more. And in that moment, I had the compromise to present my artwork. So, but the only thing I was thinking about is like, I'm here alone. I miss people. I miss touch. I miss contact. So in the end, my research took the shape of a catharsis and an installation. And what you are seeing here inside the flask, they are layers of kombucha, of bacterial cellulose, and they are connected to sensors of light. So I keep working with the idea to make a sensitive layer. Instead of using the one in the research, I make a speculation. So my pathway to still create something based on the previous research was through a speculation, and it takes the shape of a platform because I was craving for contact, and I start to see how people was afraid to touch each other because of the coronavirus. I create a platform that has a distance, a new normative distance. And when you incite light in the flasks, the kombucha, the sensors send a signal and there is a vibration in the other platform. So it's kind of sending a tactile sensation to the person in front of you. It was interesting to see people that just by coincidence, they never meet, they were in the gallery and they were sending touch to each other. So of course, this artwork talks about the haptics, the sense of touch, and also the new normality that we have right now. And we have still the idea of continuing with the second skin when we can come back to the laboratory to create a garment that is in a speculative world where we cannot touch each other because of the risk. So what I learned from this experience is that for scientists, it's very motivational to work with artists in the prototyping of new artworks, especially with biomaterials. And sometimes when you can still, you face some problems, in this case, maybe difficult results, like something didn't came as you expected, you still can use the pathway of speculation to popularize what you're doing. And the hypotheses and fabulations are not as different, like scientists and artists have an idea, test it, and then make the artwork or the paper or the product. So art and science is about making alliance between scientists, artists, and other microorganisms or biomaterials. And my takeaway from my piece was, or the most important thing was to see people make a connection through a biomaterial, in this case, the bacterial cellulose. Thank you for your time and looking forward for the discussion.
Thank you so much, Lara, for that super special talk that you just gave. I think it's super interesting her work, and I think overcoming crisis is something that we're also talking about, and how crisis has pushed forward whatever change was going to happen. Now we're moving towards that direction. There's no way that that's not the direction that we should go to, like getting more in contact with nature, collaborating with these organisms like we were talking previously. And let's see now, Orkan is gonna close up our panel with his biomaterial through soil and other stuff he's gonna talk about. Not only that, Orkan.
Hi, thanks for all this panel and all these amazing presentations. So I'm very energized with the content that I've seen. So my slides are up and let me play. Yeah, BioSummit is a very special environment for me. It's been my 4th time, I think, joining, and I am really very excited to join the community, but also share some new work, which I usually don't show before it's finalized, but this community is so welcoming that I really love the feedback and the excitement. So my name is Orkan Telhan. I am a professor, associate professor in University of Pennsylvania. And I've been working with biodesign for the past 10, 15 years, both as an educator, but also a designer working with different companies and consultancies. I also have a startup that works in the space of biotechnologies and biological design. So today I'm going to focus on some new work that is fundamentally is making me think about biodesign and biomaterials in different ways. And I hope we will have a nice conversation around these issues. So before I talk about the new work, let me tell you a little bit of where I come from. So this is the kind of work that I did 2-3 years ago together with MIT Design Lab, Puma, the sneaker and apparel company, and BioRealize, my own company. Basically, I've been very interested in the idea of embedding living organisms into materials, so the materials have some kind of agency during the life cycle of the product, whether changing the materials, changing the appearance, changing the aesthetics of the material, whether making it recyclable, biodegradable. We explore a number of different areas to really take advantage of organisms in relationship to different kinds of packaging, sneakers, apparels, sensors, but mostly by focusing on keeping them alive. This question of being alive and an alive material is important for me because when we think about materials, we always think about whether it's growing materials, whether it's using biomaterials, we always think about making something new to substitute some kind of a petrochemical process, which we all don't like anymore because it's not renewable. It has a lot of environmental impact. We try to think about materials that are better, but we don't really think about how do we design the afterlife of products. We all like to have biodegradable materials, but we don't really design for biodegradability. So my research has been really thinking about the life cycle of a product and amazing the afterlife of a product, and to see where products actually go. So today I'm going to really talk about the idea of where things go and also where things come from. And I've been really interested in lately in the idea of soil. Why soil? Because you can think about two different words that refer basically to the same thing. You can pick up a bunch of dirt from the soil and call it sand, and use it as a biomaterial in one way, or you can treat soil already as a living environment, as a material itself, without necessarily thinking of it in another context. Soil is the material, is the organism, is the life all together. The same thing when you think about the forest, you see woods or timber. When you isolate what you really want to see in this in an environment, you see only the things that you really care. But what happens if you see the soil as a material as opposed to the sand itself? So I will show you two perspectives so that you can really understand why I'm thinking of them in parallel. So even though I'm more interested in talking about soil, I'm going to start with another project that we are doing together with a big group of people. I will show you the credits in a second, but by showing you what does it mean to design with sand as opposed to designing with soil. So we've been interested in biomineralization. There's a number of research groups, designers in this crowd is also interested in biomineralization, making concrete alternatives. These are my collaborators, Dux, Ellison, Karen, and Safa. How do we think about biomineralization as a phenomena to make new kinds of materials that are not necessarily just substituting something that is out there instead of making concrete for buildings? How can you build concrete as a material that is very much like paper? It's on the surface, you coat the surface of an object, whether it's building component and a structure or architecture, so that you can think about a completely new relationship between an organism and sand to make a new kind of product. So there's a lot of, for those who know the work, know how it works, it's very straightforward, but this is a cheat slide. You basically work with S. pastöri as a bacteria, with silica, you mix it with urea and calcium, and it calcifies and turns into a solid form. We use the principle, we exploit it, but instead of growing this sand and the organism on a volume in a mold, we grow them on surfaces. A lot of hard work. Does it work? Barely works. It really is going to work, but it will take a couple of years for us to really master this. But the most important thing is that here the sand and the organisms were isolated things that never were together. We artificially brought them together as designers. We create an environment, we create a context for them so that they can really make a new kind of product. So this is some pictures from the process. We really work hard on attaching the sand and the organisms onto the surface so that the organism calcify within this special paper substrate called the vulcanized paper, which is structural, but it is not as solid as concrete. So we are trying to reinforce it and make it as strong as concrete. Okay. Does everyone see my slides okay? Yep, all right.
Yeah,
so
in a little swatch window in the corner.
Swatch window in the corner?
Yeah, like a color thing.
Okay. Really, I don't see it.
Okay, find it.
Okay. So let's move forward a little bit. These are some early experiments to see how things really grow in swatches. So these are not alive anymore, they're dried up, but these are basically concrete paper hybrids, which have sand, organisms, and things together. So the fundamental question when we were doing this research, as I mentioned, where do we get the organisms, where do we get the sand, and how do we bring them together? But it doesn't really answer the question of whether we are really creating something fundamentally interesting in terms of environmental friendliness, sustainability, or addressing the bigger issues with climate change, because ultimately we are still utilizing the same kind of design principles. Isolated things come together. We are thinking to apply, either design logic, scientific logic, or some kind of a hybrid perspective so that we really create a new kind of material. But what if you think about sand and organisms not as separate things, but something that are alive in their own environment? So I've been thinking a lot about soil, and last year I was fortunate enough to start a project in Istanbul about understanding the nature of soil in different parts of the city. So this is the map of Istanbul. You see different places, soil, there are gardens, there are different places where, you know, it's an 8,000-year-old city. There are very different places where different soil artifacts are excavated. So we are, as a team of researchers, we digged into different areas and started to analyze what kind of organisms live in this soil. This was a very rewarding process because instead of starting from an existing organism, known piece of soil, we started to really play like a detective, very much like a scientific process. You take a scoop of soil, you analyze it with genomic sequencing, you get cultures, you have no idea about them, you enrich them in different kinds of media, and then from that you start to go into different isolation methods to be able to identify them. We digged in 7 different places, 7 different gardens, and found about 7,000 different species, completely isolated. So I'll talk about patents maybe if I have time at the end, but these are organisms. When you harvest local organisms, you know, everyone works mycelium, everyone works with you know, kombucha, you know, these organisms are circulating in our vocabulary, but we have no idea what other thousands of organisms are out there. So getting a list of very new organisms from a local context was a very eye-opening process. So there's thousands of different organisms you can use for biomineralization instead of the S. pastori that I was talking about. So it's an interesting process of understanding a new way to learn about this organism culture, which can actually be very similar to really becoming, building new collaborations with new organisms which you have never ever heard about. So I'll show you a couple of different slides of how this works. You pick up a bunch of organisms, you very much like blindly try to grow them in media that you have, that is known for growing bacteria or yeast, because you have no idea how to isolate them from each other. You use different media to enrich them so that you get different phenotypes so that in which you can isolate in different media, and then prepare them grow them, if you're lucky, in liquid cultures so that you can send them to sequencing and in return learn what is in there. So why is this important? Because going back to what we do with these organisms, we looked at different ways to take organisms from the soil to design new kinds of soils as new kinds of materials that can enrich the process of composting and degradation. So instead of designing new materials with these organisms, we said, what happens if we really design new kinds of composting technologies, which utilize different combinations of these organisms? You can call them synthetic ecologies. You can call them basic composting traditions that people have been doing for, you know, thousands of years all around the world. But designing fermented composting environments, which can be customized for degrading PET, different kinds of plastics, different kinds of styrofoam by incorporating different kinds of worms. So you don't rely on the fact that one day your products are going to be eaten up by soil. You design the soil by making it more receptive for the things that we are designing as humans in these kind of projects. So I will end with this remark by saying that the idea of maybe, I think I have one more slide. This is reversed. Yeah, I will end this by saying that in this, I attended a couple of different sessions throughout the Bio Summit, and we really looked at interesting ways to bring science together, design together, art together. And there was an interesting panel on indigenous, the idea of indigenous knowledge, different kinds of knowing and making. But I would like you to think about, as part of this conversation, how as designers and artists, we can actually think about bringing different ways of knowing into our practices, whether it's rethinking our assumptions, whether we work with mycelium or working with biomineralization, what is the organism, where the organism comes from, what kind of things that we impose onto these organisms instead of building relationships with these organisms, designing a product for me is always part of a human need, human desire, human want. It's not something organisms want. But if you really want to think about a new relationship with nature, new relationship with environment, there's already a lot of knowledge that is out there that is done, studied by indigenous scholars, native scholars, and also people who have been on this planet working with different kinds of materials for thousands of years. So hopefully this will be a good way to think about our current assumptions about accessibility, because I also want this to be emphasized in this panel. The idea of not seeing the idea of accessibility as letting designers and artists going into the labs and learning how to think like scientists or engineers. There's a lot of discrepancy, asymmetry in knowledge, know-how, and tools and technologies. I never dismiss that. I work towards mitigating accessibility. But when we think about accessibility, we also level things down, we standardize things, we copy each other's practices, and we think that what we really know should be pursued in a larger community. But there's a lot of interesting things that we should think about as bringing different ways of knowing into these communities so that we can also enrich our conversations on this. Maybe there's different ways to do biodesign by not looking at science, but other kinds of knowing and making. That's how I will end. And hopefully we'll have a wonderful conversation. Thank you.
Thank you, Orkan, for your talk. Unfortunately, we don't have so much time because we have to go back to the plenary, but this was amazing. I think I'm just gonna go ahead, just highlight a few things that were mentioned before. I think that's this afterlife of materials, and when we had our talk about soil being as a black box, this magical space which is invisible, where this process happens and where materials end up transforming themselves. I think what Kai was working on, talking about this shift from standard to variation, or how variation can come into play instead of just standardizing the process. And also, if we try to replicate our current models to create this new material system, it's probably not going to work. And yesterday, Drew Andy said something that it was super interesting. He said, we are oriented to thinking about consumers, and consumers are only going to consume instead of being doers. And I think what biomaterials bring to the table is like a bottom-up approach where we can take actually this, the material making in our hands. And we have the technology, and the technology is biology in a way. So how can we harness, and how can we cooperate to do this, like Dilan was talking, to understand the system, to and then work with what you've learned about this growth process and put it into your system. I think something that is really interesting as well is nature doesn't have subtractive material method. It's adding material to generate what is done, and it's not generating excess of material. Like there is no like left out of cutting material out. And I think this shift of values and this traceability of materials and understanding where the objects that we have and the materials that we have are coming from, and that's gonna be something that would change how we behave toward objects, and also how we consume it. It's not the same to grow something and see it grow and build that relationship in a way that just buy something where you have no idea where it came from. So on this end, we want to end this panel and talking about materials in a more open view than just thinking about can I use this to make a new material? And also question, do we need a new material? And what is the fitness and where we're gonna put the material on? So thank you, everyone. I think we're 2 minutes after. This was amazing. Thank you for being here.
Thanks, Heidi. Thanks everyone for their contributions. It was lovely.